Hi, I'm Miss Sloan, and I'm just helping a few students after school reviewing photosynthesis, and we thought we'd invite you on in. So remember, we're going to do photosynthesis within the chloroplast. And the chloroplast has thylakoid membranes that are stacked up as grana, and the interior of the grana is called the lumen, and the outside is called the stroma. So I wanted you to have that so you could look right here. This is a thylakoid membrane, and across this thylakoid membrane is the electron transport chains. There are 2 electron transport chains that you need to be comfortable with, and I'll walk you through that. But the purpose of the electron transport chain is to generate both ATP and reduced NADP. So when you do the light independent reaction, that is when you're reducing CO2 down into glucose. So let's take a closer look at the light dependent reactions. So here, you should always start with kind of getting your bearings. So this is a phospholipid bilayer. These are the green membranes of the thylakoid membrane. Here's the interior of it, the lumen, and here's the outside, the stroma. We always want to start with water. Water is your source of electrons that you're going to ultimately use to reduce NADP. And these same electrons in NADP are going to be given to carbon dioxide to ultimately build sugar. Okay, put a pin in that, I'll come back to it. All right, so you need a source of electrons. You do that by breaking water in half. The oxygen is released as a gas, and the electrons are donated to the electron transport chain. The hydrogens from the water, the hydrogen ions, are going to be accumulating within the lumen in the interior of those thylakoid membranes, like think of them as hollow pancakes. These electrons are going to get excited by the sun to a higher energy level. And you're going to do a series of redox reactions through photosystem number 2. So the high energy electrons are going to go to an acceptor who's going to release it and goes to another acceptor. One of those is a molecule called PQ. PQ has the ability to grab additional hydrogen ions from the stroma and bring them into the lumen. So now I'm starting to accumulate hydrogen ions to the interior. That creates a chemiosmotic gradient, which is like water behind a dam. It's a difference in concentration because I have more hydrogen ions in the lumen than outside. It's a difference in charge because they are positive ions, and it's a difference in pH. I'm lowering the pH of the lumen. And the reason why I want to do that is so that they can move with the concentration gradient out through ATP synthase complex. This is an enzymatic protein which will catalyze the reaction of forming ATP from ADP and inorganic phosphate. This is critical because I need this energy, this ATP, to build sugar in the Calvin cycle or the light independent reaction. So just to review, the reason why we're building up these hydrogen ions, the source of them is from water, and also PQ bringing additional hydrogen ions from the stroma into the lumen. Okay, so that is photosystem 2. Photosystem 1, I excite the same 2 electrons I got from water, but this time the ride is shorter because I'm going to pass them to transfer them to NADP to reduce that NADP. Now NADP is carrying some high energy electrons that I can donate to CO2 in order to reduce it into sugar. Okay, did that cover all of that? All right, so this is the light dependent reaction. It has to precede, okay, the dark reaction, that's an old school name, or the light independent reaction, or the Calvin cycle. This Calvin cycle, when we start, we want to start with RUBP. RUBP, you can see it has 5 carbons in it. And just to demonstrate conservation of mass, we are going to say that we have 3 of them. So these are 3 C5 molecules with 5 carbons in them. That's preloaded already. You don't have to worry about that. We're going to bring in 3 carbon dioxides. Each one of those carbon dioxide has a single carbon, so we're going to say it's a C1 molecule, and we have 3 of them. We are going to snap on this CO2 onto the RUBP to give us a 6 carbon molecule. Again, 3. So we're going to say we have 3C6. That process right there is called carbon fixation. That's the first step of this Calvin cycle. We have taken CO2 that is a gas and we have made it a solid. When you bind it on here, it's very unstable. So each of these 6 carbon molecules breaks in half to C3 molecules. not a lot of energy in them. So what we're gonna do is we're gonna take the ATP that we just made in the light-dependent reaction, and we're gonna take some reduced NADP that we just made, and we're gonna use this energy and these electrons to take these from low energy to high energy G3P. Another name for that is PGAL. That's kind of an old school name. So we have a total, we had 6, we still have 6. And these 63 are very high energy. In fact, so much energy, they are the equivalent of 1/2 of a glucose molecule. Now, we are just gonna take one of those 6 away, and that's represented right here, this 1G3P. So we remove 1, 6 minus 1 is 5. So now we have 5C3, lots of high energy. What's the significance of this? I brought in 3 individual CO2s, think of them like free-range chickens, they're all over the place, they're gas, and I'm pulling out 3 carbons connected together that is a high energy 1/2 of a glucose. So 3 carbons in, 3 carbons out, each time I turn the Calvin cycle. All right, so I have 5 left. I wanna keep doing this. So what I'm gonna do to these 5 G3Ps is I'm gonna regenerate my RUBP back. That's gonna involve another investment of energy, ATP, so I can rearrange these 15 carbons in total back to another 15 carbons, right back to RUBP. I can then do this whole thing again. I'm gonna do this Calvin cycle 2 times. Each time I do it, I'm gonna invest 3 CO2s per turn. And each time I do the turn, I will pull out 1 G3P, which is a 1/2 of glucose. So if I do it twice, I will pull out 2 halves of glucose. Hope that's helpful.